All right, welcome to this explainer. Listen, if you're like most people, when you hear the word lipid or, fat, your brain probably jumps straight to diet culture. Calorie counting, stuff we're supposed to avoid, right? Well, today we are going to completely shatter that stereotype. Because far from being just the dietary bad guys, lipids are actually some of the most dynamic, multitasking, and frankly, biologically brilliant molecules in existence. By the time we wrap this up, I promise you're going to see the hidden chemistry of your own body in a completely new light. Okay, let's quickly map out our journey. We're hitting 5 key areas today. 1. The misunderstood lipid. 2. Storage, which acts as our energy batteries. 3. Structure, building the cellular fortress. 4. Signals, our chemical messengers. And finally, number 5, what happens when biology fails? Let's get right into it. Section 1. The misunderstood lipid, unifying the diversity. So what actually makes a lipid a lipid? Unlike proteins or carbs, lipids are this wildly diverse group of chemicals. They literally don't have one single structural building block. Instead, they share exactly one unifying characteristic across all of biology, their absolute insolubility in water. They just hate it. And scientists have been fascinated by these water-hating molecules for a really long time. Back in 1814, chemist Michel-Eugène Chevreul isolated this fatty substance that crystallized so beautifully it looked like mother of pearl, so he decided to name it margarine, which actually comes directly from the Greek word for pearl. It's kind of a quirky piece of history, sure, but it just goes to show how long the physical properties of these molecules have been capturing our imagination. And honestly, that fascination is entirely justified, because their diversity is just astonishing. They are absolutely not just inert blobs of fat waiting around to be burned. Lipids act as light-absorbing pigments in your eyes and in plants. They act as essential cofactors that help your enzymes function, hydrophobic anchors tying proteins to membranes, molecular chaperones, and even complex intracellular messengers. They are, without a doubt, the ultimate biological multitaskers. Moving right along to Section 2, storage, nature's energy batteries. So here's a massive biological problem. How do living organisms store immense amounts of energy efficiently without carrying around a ton of excess water weight? Well, the answer lies in fatty acids, which are these highly reduced hydrocarbon chains. But as you probably know, not all fats are created equal. Just think about the difference between butter and olive oil. Saturated fats, like the ones in butter, have 0 double bonds. They're flexible but essentially straight, which allows them to pack super tightly together into these rigid, almost crystalline arrays. That's exactly why they are solid at room temp. Unsaturated fats, on the other hand, contain cis double bonds, and this introduces a literal kink into the molecular chain. That kink prevents tight packing, keeping them fluid and liquid. Notice how the straight, saturated chains pack so neatly and predictably, while the kinked, unsaturated chains create this chaotic, loosely packed structure. It's absolutely wild to think that a microscopic kink at the atomic level literally dictates the physical properties we interact with every single day, right in our own kitchens. Now, organisms use these fatty acids to build triacylglycerols, which, simply put, are 3 fatty acids stuck to a glycerol backbone. Check out these specialized guinea pig cells called adipocytes. Look at how the cell is almost entirely consumed by this one massive fat droplet. Because lipids completely repel water, an organism doesn't have to carry the extra hydration weight that normally comes with storing carbohydrates. So these adipocytes basically act as massive backup generators. Gram for gram, they densely pack in more than twice the metabolic energy of carbs. But nature being nature has found some incredible secondary uses for this dense, oily storage. Take the sperm whale. A massive portion of its head is completely filled with spermaceti oil, which is a mix of triacylglycerides and waxes. Now, when the whale dives 2 miles deep to hunt squid, that freezing cold ocean water cools the oil. At just the right temperature, the oil crystallizes. It becomes denser, and it perfectly matches the density of the deep ocean water around it. This lets the whale basically hover effortlessly in the deep without wasting any swimming energy. How cool is that? We also see lipids forming biological waxes, which have even higher melting points. Think about bees. They construct their entire physical environment, the honeycomb, out of these waxes because they provide a nice, firm consistency and are completely 100% impervious to water. Birds do something really similar. They use waxes from their preen glands to coat their feathers, keeping them perfectly water repellent so they don't get waterlogged. All right, moving into Section 3, structure. Building the cellular fortress. Every single cell faces a really fundamental boundary problem. How in the world do you keep the inside in and the outside out when you're swimming in a watery environment?
Well, nature's incredibly elegant solution relies on membrane lipids being amphipathic. Now, this is a crucial term. It basically means these molecules have a split personality. They've got a hydrophilic or water-loving head and a hydrophobic, water-hating tail. When you toss them in water, their water-hating tails instantly panic and hide from the water by clumping together, while their water-loving heads face outward to interact with the environment. This automatically drives the formation of a double-layered sheet. It's the guarded fortress wall of the cellular membrane. Look at the visual comparison here between two very different membrane lipids, phosphatidylcholine, which is a glycerophospholipid, and sphingomyelin, a sphingolipid. If you look closely at the chemistry, they have totally different backbones, but structurally, they mimic each other flawlessly. They both present these similarly shaped tails and polar heads. It's like evolution converged on this one perfect cylindrical shape to build highly stable, completely impermeable, double-layered barriers for our cells. And right inside those membranes, we find this molecule, cholesterol. Now, I know it has a terrible reputation in the media, but we really need to redefine it here. Cholesterol is a rigid, almost flat, sterile molecule. And far from just being a dietary villain that clogs your arteries, it is an absolutely essential architectural component. It wedges itself right between the tails of other lipids to regulate the stability and flexibility of your cell membranes. Literally, without it, your cellular fortresses would just fall apart.
Okay, section 4, signals, the chemical messengers. So we're moving away from passive energy storage and static brick walls straight into the dynamic, high-speed world of cellular communication. And it really begs the question, do lipids just sit there in the membrane being boring, or do they actually talk? Well, the answer is that certain lipids are incredibly potent, rapidly firing chemical messengers. While membrane lipids usually make up about 5 to 10% of a cell's mass, a tiny fraction of specific lipids are constantly, and I mean constantly, being clipped, modified, and shipped off to send vital instructions all throughout your body. Let's look at one of the absolute most brilliant examples of this: phosphatidylinositol signaling. So step 1, an extracellular signal, like a hormone, binds to the outside of your cell. Step 2, this wakes up a molecular scissor called phospholipase C, which literally cuts the head off a specific membrane lipid. Step 3, that severed head, which is called IP3, rushes into the cell and triggers a massive, sudden release of calcium. What we are literally watching here is the physical cutting of a cell's wall being translated into an immediate, explosive cellular action. Other membrane lipids get converted into paracrine hormones called eicosanoids. These are local messengers. They don't travel systemically through the blood. They act right exactly where they're made. And they dictate some of the most powerful responses in vertebrate biology. Prostaglandins trigger things like inflammation, fever, and pain. Thromboxanes snap into action immediately to form blood clots when you get, say, a paper cut. Leukotrienes cause the smooth muscle in your lungs to contract, which, by the way, is the exact pathway that goes completely into overdrive during an asthmatic attack. So here's the crucial takeaway from all this. When you get a headache and take an everyday painkiller, a non-steroidal anti-inflammatory drug like aspirin or ibuprofen, you are directly manipulating lipid biology. That's how they work. These drugs specifically block the enzyme cyclooxygenase. By stopping that enzyme in its tracks, your body physically cannot convert the lipid arachidonic acid into those pain-causing prostaglandins. And finally, section 5, when lipid biology fails. Because lipids are so highly active, their breakdown process requires very specific enzymes to keep things running smoothly. And when this delicate biological balance is disrupted, the reality is incredibly severe. Just look closely at this electron micrograph. It's a brain cell from an infant with Tay-Sachs disease. Notice those swirling abnormal deposits in there? Those are actually complex signaling lipids called gangliosides. In Tay-Sachs, a genetic defect means the child is missing just one single specific hydrolytic enzyme, hexoaminidase A. Without it, the cell simply cannot break down the ganglioside. So it fatally accumulates in the lysosomes of the brain, leading to progressive paralysis, blindness, and tragically, early death. It's a sobering reality that underscores just how absolutely critical the rapid turnover of these lipid messengers really is to human survival. So as we wrap up our molecular journey today, I want you to remember the biochemical rule of 3. Lipids are the incredibly dense batteries storing our metabolic fuel. They're the amphipathic bricks building our cellular fortresses. And they are the rapid-fire broadcasters communicating pain, growth, and reaction across all our tissues. Far from being just fat, they are multitasking marvels that are utterly essential to life itself. Which leaves us with one final provocative question. If a molecule that we so casually dismiss on our nutrition labels is actually this fundamental to our energy, our boundaries, and our very thoughts, well, how much of our own biology are we still completely misjudging? Keep that curiosity alive, folks, and keep exploring the hidden chemistry of your own body. Thanks for joining me on this explainer.